Hello everyone, welcome to your channel Biology Spark. Today we will discuss regarding the spermatogenesis, the important topic of human reproduction. Now move to the spermatogenesis. Spermatogenesis means the process of formation of sperm. So which occurs in the gonads, male gonads, testis. Now move to the stages of spermatogenesis. Now in case of stages of spermatogenesis, it is divided into two stages, formation of spermatid and spermiogenesis. Two stages are there, first formation of spermatid and spermiogenesis. So don't confuse regarding the spermiogenesis and spermatogenesis. Spermatogenesis means whole process and spermiogenesis means spermatid to sperm cells. So formation of spermatid, spermatogonia, that is the sperm mother cells produce spermatids, and that spermatids then transform or differentiated into the well developed sperm cells, male gametes, that is spermiogenesis. So let us discuss one by one. So first we will discuss regarding the formation of spermatid. In case of formation of spermatid, now whole spermatogenesis is given here. We have a two-phase formation of spermatid and spermiogenesis. So first stage, that is formation of spermatids. Okay. Now in case of formation of spermatid, again it is divided into three phase multiplication. So let us label it as A and B. So A is a formation of spermatid. In A, again three different phase are there. First multiplication, second growth phase, and third one maturation phase. So what is multiplication phase? Now spermatogenesis is occur in testis in seminiferous tubule. The seminiferous tubule have a germinal epithelium cells. That germinal epithelium cells, they are differentiated as spermatogonia. They are differentiated as a spermatogonia. Now that spermatogonia undergoes mitosis process. 2n, particularly for example 23 into 2, 46, or we can also denote as 44 plus xy. Chromosome is occur in spermatogonia. So spermatogonia undergoes mitosis and it leads to the formation of more spermatogonia. No any change in the number of chromosome, again 44 plus xy, because mitosis is there, only multiplication, increase in the number of spermatogonia. Now this spermatogonia undergoes growth phase. So move to the second phase, growth phase. What is the growth phase? Where spermatogonia increase in the size, they become large. They called as primary spermatocyte. So when this spermatogonia increase in the size, they become enlarged. That is called as a growth phase. And the cells after growth phase is called as a primary spermatocyte. Again the same number of chromosome, 44 plus xy. So they are the primary spermatocyte. This primary spermatocyte now undergoes meiosis I. So we know the meiosis is a reductional division, and in case of meiosis I and meiosis II, meiosis I is a reductional division, but meiosis II is a equational division. Reductional means the number of chromosome become half is that of parent cell. Now we know that primary spermatocyte is a 2n, diploid, 2n, two set of chromosome, 44 plus xy is there. After meiosis, the number of chromosome become half is that of parent cell. So 46 leads to the 23. So after meiosis I, it is called as a secondary spermatocyte. The number of chromosome become 23. So one secondary spermatocyte have 22 plus x, while other have 22 plus y. So two different spermatocyte, one which have a X chromosome, other which have a Y chromosome. So now we are in a third phase, that is maturation phase. So maturation phase involve meiosis process. Maturation phase have meiosis process. At the end of meiosis I, it is called as a secondary spermatocyte. This secondary spermatocyte undergoes meiosis II, complete its second meiosis. We know the second meiosis is similar to the mitosis, equational division. So no change in the number of chromosome, and the cells which are formed at the end of meiosis II is spermatid. That spermatid have the same number of chromosome, same set of chromosome as that of secondary spermatocyte. So 22 plus x, again the same number is there, 22 plus x, 22 plus x means total 23. 22 plus y, 22 plus y, same as that of secondary spermatocyte. So that is a spermatid. So 4 spermatid are formed from the one primary spermatocyte or from 2 secondary spermatocyte, or we can say that at the end of meiosis process 4 spermatid are formed from one primary spermatocyte. So this is the first stage of spermatogenesis, formation of spermatid. Again 3 phase, multiplication, growth and maturation phase. Now these spermatids are converted into spermatozoa, sperm cells. They are differentiated, morphologically they undergoes differentiation. Means this spermatid start to differentiate into the morphological changes like formation of head, tail, middle piece, centriole, and that is called as a sperm cells or spermatozoa. The process of formation of sperm cells from spermatid is called as a spermiogenesis, and that is the second stage of spermatogenesis. So ultimately at the end of spermatogenesis, from one spermatogonia or either from one primary spermatocyte, we have a 4 number of sperm cells. All are active, all are functionally similar, all are able to undergo fertilization. So again, now as we have already discussed that germinal epithelium, so here the testis internal structure, seminiferous tubule is given. Now that seminiferous tubule, it have a diploid spermatogonia. It carry out the mitotic proliferation, growth phase, multiplication and increase in the cell number. So there is a more and more number of spermatogonia which are increase in the size, that is called as a primary spermatocyte. That spermatocyte entered into the meiosis. But at the end of meiosis, we have a meiosis I, we have a secondary spermatocyte, and at the end of meiosis II, the haploid round spermatid is there. So spermatids are round in shape, while sperm cells, the body is differentiated, the cell is differentiated into head, middle piece and tail like structure, that is called as spermatozoa. So that is cytodifferentiation. Cytodifferentiation means morphological change is occurs in the spermatid that leads to the formation of spermatogonia or spermatozoa. So now spermatogenesis, we have already completed the whole stages, but in our seminiferous, in the structure of testis, seminiferous tubules have a various cells, germinal epithelium cells, then which is called as a spermatogonia, which are differentiated as spermatogonia. This spermatogonia undergoes primary spermatocyte, increase in the size, this becomes secondary spermatocytes, then it becomes spermatid and spermatozoa. So seminiferous tubule, we know the structure of testis which have so many testicular lobules, around 250 lobules are present in one testis. That lobules consist of 1 to 3 seminiferous tubule in each lobule. In each lobules 1 to 3 semini. So on an average 750 to 1000 seminiferous tubules are present in each testis, and spermatogenesis is occur in a seminiferous tubule. This is the lumen of seminiferous tubule, and sperm cells are released in the lumen of seminiferous tubule. Then they are released into the rete testis from seminiferous tubule, that is called as spermiation. So here the summary is there: spermatogonia, mitosis, primary spermatocyte, meiosis I, secondary spermatocyte, meiosis II, spermatid, spermiogenesis, that is formation of spermatozoa. So overall, the spermatogenesis, you have to remember that a primary spermatocyte, one primary spermatocyte, it leads to the formation of two secondary spermatocytes. One secondary spermatocyte, two spermatids are formed from one secondary spermatocyte. Ultimately, one primary spermatocytes give four different sperm cells. So from one primary spermatocytes, four sperm cells are there. So in NEET examination, the questions are asked like, for formation of 400 sperm cells, how many primary spermatocytes are required? How many primary spermatocytes are required? So 4 spermatocytes. So we have to divide by 4. Ultimately, 100 primary spermatocytes give rise to the 400 sperm cells. Or how many secondary spermatocytes are required? So we have to divide by 2. So 200 secondary spermatocyte, which leads to the formation of 400 sperm cell. Secondary spermatocyte, which gives 400 sperm cells. Secondary spermatocyte is haploid and primary spermatocyte is 2 and diploid. So the ploidy of spermatocyte, primary is 2n and secondary is haploid always. So this is the overview of spermatogenesis. Hope you enjoy this video. So don't forget to like, share and subscribe my channel.